So first you start by establishing a correlation, which we have done. And right now what we're doing is that we are actually recruiting patients with Parkinson's disease that are at different times from disease diagnosis. And this, we think, will reflect disease severity. And so what we're trying to do is to see if the levels of oligomeric alpha-synuclein are correlated with time from disease diagnosis. So we're going to try to get a sense of the disease severity. We also are eventually planning to try to test this in patients without a diagnosis, as well as patients who exhibit diseases that overlap with Parkinson's disease, such as patients with atypical Parkinsonian syndromes, and patients with other neurological disorders, of course. So to determine how specific this may be, and also whether this has predictive ability going forward if a patient is at an early stage of Parkinson's disease. This project is very much a team effort. We have a whole team at the University of Southern California, including colleagues in neurology and pharmacy and preventive medicine. And so we work very closely with Mark Liu in neurology, who is a neurologist and who is very excited about the potential for applying this in his clinic. And so I think that you know, the part of the passion for the project is driven by the clinical need that I sense from my neurology colleagues and their frustration that they don't have better ways to diagnose patients at early disease states.